All right, let's begin talking about mitosis and cell division. Now, before we begin, there's some things you need to remember. Now, cell division and cell growth causes organisms to become larger. When a cell becomes too big, they start cell division instead of continuing to grow. When an organism stops growing, cell division is still going on because you're always going to have cells that are going to die and need to be replaced. But it's important to remember that cell division will always continue even when an organism, regardless of what it is, stops growing and developing. Okay, now the first thing that must happen is a cell must copy all of its genetic information. That's the very first thing that has to happen before a cell divides. Now, if it didn't, the results would be disastrous. It wouldn't have all the instructions to do what it needs to do. All the genetic information is packed into what we call chromosomes. We know chromosomes are found inside the DNA, and the DNA is found inside the nucleus of a organism, with the exceptions of prokaryotes. Now, even prokaryotes, such as E. coli, they have genetic information too. The DNA molecule of E. coli is about 1.6 millimeters, and that's about 1,000 times larger than the cell itself. So DNA is very, very large, but it's compacted so close together in a tight space. Now, to put this on a scale that's easy to understand, imagine taking a 300-meter rope, or which is about 900 feet, and packing it into a book bag. You'd have to make that rope so tight and so compact to actually get it in there. And that's what cells do with the DNA. Now, we said in eukaryotes, we said the chromosomes are found inside the DNA, which is found in the nucleus. Now, in prokaryotes, all the chromosomes are found inside the cytoplasm because it doesn't have a nuclear membrane-bound organelle. Now, eukaryotic chromosomes. Now, eukaryotic organisms are much more complex and contain more than one chromosome in each cell. For example, humans have 46 chromosomes, fruit flies have 8 chromosomes, carrots have 18. In each and every cell, you're going to find those amount of chromosomes. Now, chromosomes form a close association with histone, which is a protein. Now, the complexity of histone and chromosomes is called chromatin. So you'll see that word chromatin, and when you want to know what that is, it's histone and chromosome. It's the complex of histones and chromosomes bounded together. Now, the histone and DNA form bead-like structures during the prep for cell division. Now, these bead-like structures are called nucleosomes, and we'll take a look at what these pictures of these in just a moment, so it's a little easier to understand. Now, nucleosomes pack together even tighter during cell division. The cell only has so much space, and it doesn't want to rupture and burst, so it's going to pack together very, very tightly. Now, the reason cells go far to package their DNA, the reason they go out of their way to package that DNA, is because it allows for equal division of DNA. Now, chromosomes make it possible to separate DNA precisely during cell division. So let's take a look at this picture. So here we have a centromere here in the center. We'll be looking at that again. We have a duplicated chromosome. We have chromatids. Now in here, if we zoom in really close, we can find all these thread-like structures, and they're bound and wound tight together, and we call these supercoils. All right, if you unwind this a little bit, you'll see the supercoils. Kind of looks like a telephone. Kind of looks like a telephone cord here. It's wound very tight together, and when you unwind it a little more, you'll just see coils, and you see these little bead-like structures here that are wound tightly together. When you unwind it a little more, you see that these bead-like structures are actually the nucleosomes. And the nucleosome, which is this whole thing, each of these beads are the histone proteins, and they're packed together to form nucleosomes. Now, when you unwind this thread even more, you'll actually find the DNA. So go ahead and take a look at this. Review the nucleosomes, the chromatids, the centromere, the histone proteins. We'll be talking about all this more in just a few moments. Okay, so the prokaryotic cell cycle. Well, prokaryotes, which are mainly going to be your bacteria, have a regular pattern of growth. Now, rapid DNA replication and cell division will occur when the conditions are ideal. If the conditions are almost perfect, you're going to have rapid DNA replication and cell division. Now, the DNA begins to copy, and then the cell begins to divide. Now, this is what we call asexual reproduction, which is also known as binary fission. Asexual, A meaning without sex cells or without gametes. No sperm and egg here, just it's able to do it on its own just by replicating itself and splitting. So that's asexual reproduction or binary fission. Now, when the DNA is replicated, it attaches to different regions of the cell membrane. Fibers form from one side of the cell to the other side from the DNA. The fibers constrict the cell further and it pinches inward. Now, this pinching inward results into two perfectly replicated cells. It's identical to the cell it's being produced from, and you have two identical cells. That is asexual. There's no partner needed here. It can do it on its own. Now, eukaryotes. Now, this consists of four phases, G1, S, G2, and M. Now, there's a lot going on here, and if you break it down into sections, it's a lot easier to understand. Now, each phase in the entire process depends completely on the cell. Interphase is the in-between time in which the cell grows. Now, interphase contains the phases G1, S, and G2. So when you look at the four phases here, you see G1, S, and G2, and M. You're like, oh my God, this is going to be a lot. But look right here. Interphase is G1, S, and G2. So let's talk about what those mean. Now, in G1, cells do most of their growing in this phase. Cells increase in size and synthesize new proteins and organelles. And the G stands for gap, which is misleading. It's wrong today because it isn't a gap of activity, but it's a lot of growing. Now, gap was used in the early research. Then we have what's called the S, which stands for the synthesis phase. New DNA is synthesized when chromosomes are replicated. At the end of the S phase, the cell contains twice as much DNA as it did at the beginning. And that's because the DNA and the chromosomes have been replicated. Well, if it's been replicated and copied, of course it's going to contain twice as much DNA as it did in the beginning. Then we have G2, which is the shortest phase of interphase, because remember, interphase contains G1, S, and G2. Now, many organelles required for cell division are going to be produced here. Now, then we have the M, because remember, it consists of four phases, M, which produces two daughter cells, and that M stands for mitosis, and this is when the cell divides. There are two stages of cell division. There is mitosis and cytokinesis. And notice we say cell division, not cell replication. So here we're talking about cell replication in the cell cycle, and then we have cell division when the cell actually divides, which is mitosis and cytokinesis. So mitosis is when the nucleus divides, and then cytokinesis is when the cytoplasm divide. And these two overlap. So let's talk about mitosis right now. So we have G1, S, and G2. Then we have mitosis, which consists of four phases. And if you can remember this anagram here, PMAT, that stands for prophase, metaphase, anaphase, telophase, is actually an acronym. Prophase, metaphase, anaphase, telophase. Now, mitosis can last several minutes to several days. It depends on the type of cell that's being produced. Now, prophase is the longest and can take up to half the time of mitosis. Prophase is probably not the most difficult to learn, but it's one that, there's a lot going on. And once you get prophase down, the rest will come super easy. Now, DNA material condenses and chromosomes become visible. You can actually see them. The outside the nucleus, a spindle starts to form. We'll look at pictures in just a moment. Now, a DNA molecule attached along their length at an area called the centromere, and we'll talk about what that means. Each DNA strand is what we call a chromatid. Now, in that chromatin, a spindle, which is made of microtubules, begins to form, which will help separate the duplicated chromosomes. You may be saying, you lost me. I don't know what that means. Don't worry, we'll look at a picture in a few moments. Now, we have centrioles, which are duplicated during interphase, and then begin to move to opposite ends of the cell. At the end, the chromosome coil more tightly, the nucleolus disappears, and the nuclear envelope begins to break down. Now, we're going to talk about these spindles, we're going to talk about these centrioles, and we're going to talk about these centromeres in just a moment. So stick with me here. So let's move on to metaphase, meta meaning middle, and it's generally going to be the shortest. It's the centromeres of the duplicated chromosomes line up across the center of the cell. So we have meta, which means middle or center, and that's why it's called metaphase, because the chromosomes are lining up across the center of the cell. So spindle fibers connect to the centromere of each chromosome to opposite ends of the cell. So here we have these words again. We're going to take a look at a picture, don't worry. Then we have anaphase. So remember PMAT, prophase, metaphase. Now we're on anaphase. Begins when the sister chromatids begin to move apart. The chromosomes separate and move along the spindle fibers to opposite ends of the cell. Now, if you have no idea what we're talking about, let's take a look at a picture. So this is metaphase. I know it's metaphase because when I look at these chromosomes, they're lining up across the center of the cell. And right here in the center, we have what's called the centromeres. All right, they're lining up right here. Now, the spindle fibers are going to connect from the centrioles to the centromeres, and they're going to start connecting, and then they're going to pull them apart. This is metaphase because they haven't started pulling them apart just yet. All right, so when we have the centromere, we're talking about right here in the center. But then anaphase, we have the centrioles that have connected to centromeres, and they're pulling these chromosomes apart to opposite ends. So in prophase, we have these centrioles moving to opposite ends of the cell. So here's the centriole. It's moved to one end. The other has moved to the other end. Then they're going to send out these spindle fibers, connect to the centromere, which is going to be right here in the middle. We can see they're doing that. It's going to start pulling these sister chromosomes, or these duplicated chromosomes, apart. All right, let's move on to telophase, which is the final phase of mitosis. Remember PMAT, prophase, metaphase, anaphase, telophase. The chromosomes, which were distinct and condensed, spread out into a tangle of chromatin and becomes a nice tangle. Now, the nuclear envelope forms around each cluster of chromosomes. The spindle breaks apart, and the nucleolus forms in each daughter nucleus. So when we look back here at this picture, we're going to have two cells that begin to replicate. And then we're going to have, this is going to become the nucleolus, and we're going to have an envelope form, and we're going to have an envelope form around this one once it gets so close together. Now, we said that. So mitosis is complete, but it has one more stage to go. Remember, mitosis is PMAT, but there's one more stage, and that's cell division, which is mitosis and cytokinesis. Now, cytokinesis is when the cell is actually going to split. Now, as a result of mitosis, two nuclei, each with a duplicate set of chromosomes, are formed. In phase of the cell cycle, the G1, the S, the G2, and the M, remember, the M phase of that is cytokinesis, the division of the cytoplasm. It usually occurs around the same time as telophase. Notice it says around the same time, not the same process. All right, we'll pause here. All right, to the last part. So we've talked about cell division in prokaryotic organisms. Let's talk about eukaryotics, which we've been talking about in animal cells and plant cells, the final stage, cytokinesis. So in animal cells, cell membrane is drawn inward until the cytoplasm is pinched into two equal parts. So here we have an animal cell. This is a real animal cell. We can see the membrane is starting to move inward. It's starting to pinch together, and it will separate. Now each cell or each part contains its own nucleus and its own organelles. So here we're going to have a nucleus that's going to start to form. It's going to pinch off, and this cell is going to go one way. This cell is going to go another. We'll have two perfectly replicated cells. So we can see it's starting to pinch off and break off here. Now, in plant cells, the cell wall, which is very rigid, it prevents the cell membrane from being flexible. A structure known as a cell plate, and if we look close, we can see the cell plate forming right here, forms between the divided nuclei. So here we have a nucleus. Here we have a nucleus. It starts dividing between these nuclei right here. Now, the cell plate eventually becomes the membrane. And remember, plant cells have not just a cell membrane, but also a cell wall. And the cell wall forms around the membranes, completing the process. So that is the introduction to mitosis and cell division. If you have any questions, you can always post a comment below or send us an email.